Plant-based compounds, particularly polyphenol like resveratrol and curcumin, are attracting growing scientific interest for their potential to slow cellular aging and support telomere maintenance through finely tuned biochemical pathways. These naturally occurring substances, found in foods like grapes, turmeric, berries, green tea, and dark chocolate, have long been praised for their antioxidant and anti-inflammatory properties. But recent research has begun to uncover their more targeted roles in activating cellular stress response systems, influencing gene expression, and potentially enhancing telomere stability by interacting with telomerase and DNA repair proteins. Resveratrol, a stilbene compound most famously found in red grapes and red wine, is perhaps the most studied plant polyphenol in the context of aging. One of its key molecular actions is the activation of SIRT1, a member of the sirtuin family of proteins. Sirtuins are NAD-dependent enzymes that help regulate cellular stress responses, DNA repair, and metabolic health. SIRT1, in particular, has been shown to influence telomeric stability by enhancing chromatin structure and protecting telomeric regions from degradation. It may also support the activity of telomerase, the enzyme responsible for rebuilding telomeres in certain cells, although this effect appears to be context-specific and may require synergistic signals from other cellular pathways. By promoting a genomic environment that favors longevity and DNA repair, resveratrol stands out as a potentially powerful, low-toxicity agent for telomere support. Curcumin, the primary bioactive compound in turmeric, offers a complementary set of benefits. While it does not activate sirtuins directly, curcumin is a strong modulator of inflammation, oxidative stress, and gene expression. It has been shown in vitro to reduce pro-inflammatory cytokines such as TNF-A and IL-1S, downregulate NF-B signaling, a major pathway involved in chronic inflammation, and enhance the activity of antioxidant enzymes like catalase and glutathione peroxidase. These anti-inflammatory and antioxidative effects create a biochemical environment conducive to preserving telomeres. In several cell culture studies, curcumin has demonstrated telomere protective properties, including the attenuation of telomere shortening in cells exposed to oxidative stress. While these findings are early stage, they support the hypothesis that curcumin may help reduce the cellular wear and tear that accelerates telomere loss. Beyond resveratrol and curcumin, other plant-based polyphenols, such as epigallocatechin gallate, EGSG, from green tea, quercetin from onions and apples, and apigenin from parsley and chamomile, have also shown promise in influencing cellular stress pathways relevant to telomere biology. These compounds often exhibit overlapping activities, including scavenging free radicals, modulating inflammatory responses, improving mitochondrial function, and enhancing DNA repair capacity. The combined impact of these mechanisms may not only stabilize telomeres, but also improve the broader genomic integrity required for long-term cellular resilience. A particularly intriguing aspect of plant-based compounds is their hormetic effect, the capacity to mildly stress cells in a way that stimulates adaptive, protective responses. Many polyphenols function as mild pro-oxidants in low doses, triggering the upregulation of endogenous defense systems. This controlled stress appears to strengthen the cell's ability to respond to more damaging insults, improving antioxidant response, DNA repair efficiency, and metabolic stability. Such hormetic effects are increasingly recognized as a key feature of effective longevity interventions, and telomere preservation may be one of their downstream benefits. Of course, translating in vitro or animal model results into human health outcomes remains a challenge. Bioavailability, the ability of these compounds to reach cells in active form, is often low, particularly for curcumin, which is rapidly metabolized and poorly absorbed on its own. However, strategies like combining curcumin with piperine from black pepper or using lipid-based delivery systems have been shown to significantly enhance its uptake. Likewise, resveratrol's effects may depend on dose, timing, and the presence of other nutrients or cellular conditions that influence sirtuin activity and NAD levels. Still, the relatively low toxicity, broad availability, and pleiotropic benefits of these plant compounds make them a promising part of a comprehensive telomere protection strategy. When consumed regularly as part of a balanced, antioxidant-rich diet, They may help reinforce cellular defenses against the everyday stresses that accelerate biological aging. While more clinical trials are needed to confirm their direct effects on human telomere dynamics, the mechanistic rationale is strong, and the overall impact on inflammation, oxidative stress, and genomic stability makes them valuable allies in any effort to promote healthy aging. In a world increasingly seeking natural and sustainable longevity tools, plant-based polyphenol represent a scientifically grounded, nature-derived approach to supporting telomere health and enhancing our internal resilience. They don't just promise longer life. They support the foundational systems that allow us to age with greater strength, clarity, and vitality.